When a patient takes a drug along with an enzyme-inducing drug, the first and most important event is increased synthesis of liver microsomal enzymes. Because of this, the liver starts working faster than normal. As a result, other drugs are metabolized more quickly, which leads to a shorter duration of action and sometimes therapeutic failure. A classic example is oral contraceptive pills failing when given with rifampicin, as rifampicin, which is enzyme inducer, result in increased the metabolism of contraceptive pills. At the same time, faster metabolism can sometimes produce more toxic metabolites, which explains why paracetamol toxicity becomes more likely in patients taking enzyme inducers. Paracetamol is metabolized into a highly toxic compound called N-acetyl P-benzoquinone imine. Under normal conditions, this toxic metabolite is immediately neutralized by glutathione and causes no harm. But in situations such as overdose, chronic alcohol intake, or enzyme induction, excessive NAPQI is produced and glutathione gets depleted. Once glutathione is exhausted, NAPQI starts binding to liver cell proteins, leading to hepatocellular injury and liver necrosis. Enzyme induction also increases the breakdown of vitamin D, leading to vitamin D deficiency, and over long periods, this can result in bone diseases such as rickets in children and osteomalacia in adults. Another important point is autoinduction, where a drug increases the metabolism of itself. For example, carbamazepine induces its own metabolism, so over time, a higher dose is required to maintain the same therapeutic effect. The response to drugs can also become unpredictable in certain populations like smokers and chronic alcoholics because their liver enzymes are already induced. Finally, enzyme induction can sometimes be used for our benefit, not just as a problem. A classic example is phenobarbital in neonatal jaundice. In newborns, the liver enzymes that conjugate bilirubin are immature and work slowly, so bilirubin accumulates and jaundice appears. When we give phenobarbital, it induces these liver enzymes, making them work faster. As a result, bilirubin is conjugated more efficiently, cleared from the body more quickly, and the jaundice improves.